Good morning, everyone.
Morning.
Welcome back. Hope you're doing okay this morning. Okay, so well, we're getting dangerously close to Thanksgiving, aren't we? So counting today's lecture, you have 6 lectures left for the entire semester, right? Woohoo! We're almost there, okay? Hang in there. Okay, so we're going to get back into chapter 12 this morning, really start to get into the meat of the nerve impulse physiology, okay? Do you have any questions right now about content or our semester schedule? Okay, since everyone's quiet, let's see what we can do here. I'm going to share my screen with you and get started here. Okay, so welcome back, gang, and we'll get back into chapter 12 here. At the top of the note-taking document, I put some reference to Wiley Plus support videos that are important for understanding nerve impulse physiology. My intention is to show both of those videos to you today, but if I don't get to all of them, I want you to be aware of them, right? In your Wiley Plus online course resources, right under the 3D physiology, you have these 2 videos, membrane potentials, excitable cells, and the resting membrane potential, and membrane potentials graded in action potentials. Both of these are very good videos, and they can support your learning through this chapter, okay? So just be aware of those, and you might want to take a second look at them during your private study time over the next few weeks. Okay, so we'll be showing you at least one of those videos today, hopefully both of them. From last time, we were talking about neuroglia of the central nervous system, and we had just got done defining some of the functions of astrocytes. So once again, we're looking at figure 12.6, in my textbook, that's page 427. So let's continue that conversation. The second type of neuroglial cell in the central nervous system that we'll put down in your notes is the oligodendrocyte. Okay. Oligodendrocytes are significant because these are the types of cells that form myelin sheath in the central nervous system. I have definitely encountered students in the past that are aware of myelin sheath, but you tend, for some reason, folks tend to think that that's only a peripheral nervous system thing, and that's not quite true. There is myelin sheath in different areas of the central nervous system, but its composition is a little bit different from the myelin sheath you see in the peripheral nervous system. At any rate, the oligodendrocyte is the type of cell, it's a glial cell in the central nervous system that forms and maintains myelin sheath in the central nervous system. We'll talk later about what the overall purpose of myelin sheath is, but just to give you an idea to think about at this moment, generally speaking, myelin sheath does a number of things for the nervous system, but I think probably the most significant thing is that myelin sheath is capable of accelerating the transmission speed of the electrical signal. So the electrical signals cover the same distance faster when the axons are myelinated than when they are not myelinated. Okay. All right, folks, so that's oligodendrocyte. A third category of neuroglial cell in the central nervous system are microglia. Okay. Microglia function primarily as phagocytes, so they are a type basically a type of white blood cell that's participating in body defense. They have a role to play in defending our bodies against microorganisms that might invade the brain and spinal cord. And then the fourth type of neuroglial cell that we'll document in your notes from figure 12.6 are in fact the ependymal cells, kind of a strange word, okay, ependymal cells. The ependymal cells line the ventricles of the brain. So you remember in lab, we identified the lateral ventricle and the third ventricle in the visible body software, right? It was sort of that light blue, somewhat translucent structures that we showed. Third ventricle was immediately between the left and right hemispheres, the lateral ventricle. So on both the left and right hemispheres, it's a weird C-shaped structure with a spine coming off the back. At any rate, the ventricles of the brain are, they're made of many cells, but the inner lining of the ventricles is made of ependymal cells. So ependymal cells line the ventricles of the brain and the central canal of the spinal cord that is filled with cerebrospinal fluid. The purpose of the ependymal cells, as best as we can tell from a scientific perspective, is to produce and circulate the cerebrospinal fluid. So this lining of cells in the ventricle and in the central canal of the spinal cord actually helps filter blood plasma to create the cerebrospinal fluid. Okay. All right, folks, any questions right now? Okay, let's continue then. So we're going to shift gears here just a little bit and talk to you about neuroglia of the peripheral nervous system. Okay, and so for this, I suppose the best picture you could look at would be figure 12.7. So just turn the page in your textbook. You should find figure 12.7. Neuroglia of the peripheral nervous system. There's not quite as many cells that we need to define here. So the first category, and probably the most prominent category, is the famous Schwann cell. Schwann cell, that's S-C-H-W-A-N-N, Schwann cells. Okay. Schwann cells form the myelin sheath within the peripheral nervous system. So Schwann cells are comparable to, they are analogous to the oligodendrocytes of the central nervous system. Form the myelin sheath within PNS. These cells are analogous to the oligodendrocytes of the central nervous system. Okay, so Schwann cells, they do a number of things. What I will say is this, that Schwann cells have been shown to help regenerate damaged regions of axons. So if an axon is damaged from for whatever reason, through inflammation, infection, physical trauma, through a physical injury, like a compressing injury at You might be working in a heavy industry or something like that, and you get a partial crushing injury. Well, Schwann cells can sit down on sort of the broken area of the axon and help mediate its healing and its rejuvenation and its repair. So it helps, Schwann cells help regenerate damaged sections of neurons. Okay. In the context of myelin sheath, let's talk about this. In the context of myelin sheath, what do Schwann cells do? Okay. All right. So let's talk about this a little bit. Okay. So another way of phrasing my question is, what is the function of myelin sheath here? Okay. All right. So the function of myelin sheath, both in the central nervous system and in the peripheral nervous system, is to basically accomplish several goals. Okay. First off, if you take a look at figures 12.7 and figures 12.8, you will see the myelin sheath. Okay, so myelin sheath is when you take a Schwann cell and you wrap it around multiple times on top of itself. Okay, so let's start here. Okay, remember that myelin sheath is a multi-layered wrapping of a Schwann cell's plasma membrane, or equivalently an oligodendrocyte's plasma membrane in the central nervous system, around an axon. Okay, and let me highlight something for you here, folks. Okay, myelin sheath is not really myelin sheath until it is a multi-layered wrapping. Okay, so let me give you an analogy. Okay, let's imagine that your finger is an axon and that we have a band-aid because you've cut your finger. Okay, okay. Okay. How in the world do you spell Band-Aid these days? Help me out, folks. How do I spell Band-Aid in the chat window? Band-Aid.
Bandaid.
Thank you so much. All right. Something's still weird about that. At any rate, a finger and a Band-Aid. Okay. So if you take the Band-Aid and you wrap it around the finger one time without overlapping any of the Band-Aid on top of itself, then technically that is not considered myelin sheath. If you had a very large Band-Aid, and you wrapped the Band-Aid around the finger, and because the Band-Aid was so long, some of the Band-Aid continued to wrap a second time and layered on top of itself, so we had multiple layers of thickness of the Band-Aid, then that would be myelin sheath. Christian, thank you for the spelling help there. Appreciate that. Okay, so wrapping a Schwann cell, we'll do it this way. Wrapping a Band-Aid once around the finger is not myelin. wrapping a Band-Aid multiple times on top of itself around the finger is myelin. Okay. All right. So let's see. I'm going to reference figure 12.8 here as we talk about this. Okay. All right. So I know that's a little bit wordy, but I hope you're getting what I'm saying here. Okay. If you have multiple layers of a cell wrapped on top of itself many times, that's myelin sheath. But if you only have one layer and it doesn't overlap itself, that's technically not myelin sheath. Okay. And so let's continue our conversation here. Okay. So the functions of myelin sheath are several-fold. Okay. Myelin sheath functions to first act as an electrical insulating tape. And the reason for that is, remember, when we're talking about axons, we're typically talking about axons that are part of a larger structure that we defined last time called a nerve, right? And so those axons are running parallel to each other, okay? And so those axons are stacked very close to each other. There's very little physical separation between them. So if I could just do a little bit of drawing, okay, just a little bit of drawing here. So if we have a typical nerve fiber, we're going to have multiple axons running parallel to each other like this. Okay, and so they would be stacked inside a nerve. So these are multiple axons, right? And so the idea here is that, you know, an axon is going to be propagating an electrical signal. So I'll just indicate that with this little red thing here. Because the separation of space between any two axons is very thin, there's not a lot of distance because the axons are packed together very tightly, there is a danger that in some cases the electrical signal can actually cross over to another axon that's next door. You wouldn't want that to happen because that means the electrical signal is going to the wrong destination. Okay, so the presence of myelin sheath, if I take an axon and I start wrapping it in myelin, if I start wrapping it in myelin, then that prevents the jumping of the electrical signal to a neighboring axon. It prevents that because of the insulation properties of the plasma membrane of the Schwann cell wrapped around the axon. Okay, so quick question in the chat window, folks. Am I making sense? Is this conversation okay right now? Okay, all right, anything you'd like to ask? All right, okay, folks, thanks for your feedback, I appreciate it. So one of the functions of myelin sheath, which is formed by a multi-layered wrapping of a Schwann cell across the axon, is to prevent the electrical short circuiting. Okay, so let me just finish my statement here. Prevent jumping of electrical signals. Okay, let's continue. A second function is to accelerate the transmission speed of the electrical signal. Okay, this is a phenomenon called saltatory conduction. Okay, so I am quite sure if you did a YouTube search for saltatory conduction, you could find some useful videos on this. Okay, and some of the videos I might show you today might tickle you a little bit with the idea of saltatory conduction, although that's probably not the main goal of the videos I will show today. So here's the analogy. Here's the analogy I would use with you. Okay, and you know, it's going to take a while going through this chapter, hearing all the pieces of factual information you need to hear before you can come back and review and say, okay, now I can put it all together, right? It's going to take a while to synthesize all of this. But with saltatory conduction, it's kind of like you're playing a game of basketball. Let's think about that for just a moment. If we're all sitting on a basketball court, and let's say that we have basketball hoops at each end of the court, right? And so let's say that you're on my team and we are far away from our hoop at the other end of the court. Okay, so you're on my team and we are far away from the hoop at the other end of the court. Let's say that I have the basketball. Okay, if I have the basketball, there are three things I could potentially do to make a hoop. Number one, I could take the basketball and I could throw it like a Hail Mary pass at the end of the game and just hope it gets in. That never happens in the body, so that kind of analogy is not accurate at all. So just throw that idea out the window. Your body does not throw an electrical signal down the court. It doesn't do that. The second thing you could do, or I could do if I have the basketball, is I could literally dribble the basketball the whole way down the court till I'm right underneath the net, and then I could take the shot for myself. Folks, if you take that kind of behavior, if you or I do that, we dribble the ball down the entire court till we get to the net, then take the shot. That is in analogous terms, that would be similar to an unmyelinated neuron doing what it does to spread the electrical signal. Okay, so let me try to get this into your notes here. Playing basketball. Okay. Dribbling the basketball down the entire court to make the shot. Okay. Dribbling the basketball down the entire court to make the shot is moving the electrical signal down an unmyelinated neuron. So the alternative in our basketball analogy is if you or I have the ball and the courts and the hoops down at the other end, if I'm not going to do a Hail Mary, which never exists, and I'm not going to dribble the ball down the court, my really only other option is to take the ball and pass it to a teammate that is closer to the net already. Okay, so a bounce pass in basketball, right, where you push the ball away from yourself, you bounce it off the floor, and a teammate picks it up, and the teammate is already closer to the net. Well, folks, bounce passing the basketball, let's just say for folks that are not comfortable with basketball, simply passing the ball to a teammate that's already closer to the net is similar to saltatory conduction. Okay. Passing the basketball to a teammate that is physically closer to the net is saltatory conduction. Okay. This saltatory conduction of passing the basketball, what I'm suggesting is we're not passing the basketball, we are passing the electrical signal. We are passing the electrical signal. The only reason this happens is because of the myelin sheath. The myelin sheath enables the passing of the electrical signal. This only happens in myelinated neurons. Okay, so myelinated neurons make saltatory conduction possible. Okay. All right, folks. So, I want to encourage you to do some YouTube searching for saltatory conduction. I think if you watch a video to it will become clear what we're trying to say here. Do you have any questions right now? Okay, so the third function of the Schwann cells is to act as a glial cell. And what I'm saying is, even though we're calling it a Schwann cell and we're saying the Schwann cells form the myelin sheath, even in the context of the myelin sheath, the Schwann cell is a glial cell. And so what is one of the general purposes of all glial cells? Well, it is to support, protect, and help nourish the neurons. Okay, so functions of myelin sheath, once again, act as electrical insulating tape to prevent this short circuiting or the jumping of the electrical signal from one axon to the next. We don't want that to happen. So the myelin sheath helps prevent that. 2, to make the electrical signal travel the same distance faster in the presence of myelin sheath. So myelin sheath effectively accelerates the transmission speed of the electrical signal. That's like passing a ball to your teammate who's standing underneath the basketball net. And finally, third thing is to support, protect, and nourish the neurons of the nervous system. Okay, folks, any questions about the myelin sheath? Okay, so the topic of myelin sheath came up here in our description of the Schwann cells. Typically, we talk about myelin sheath with the Schwann cells in the peripheral nervous system more so than the oligodendrocytes in the central nervous system, because as you've just heard, one of the functions of the Schwann cells as part of the myelin sheath is to accelerate the transmission speed of the electrical signal. So if you have an electrical signal that needs to spread down from your brain, down your spinal cord, down the length of your entire leg to your big toe, that is a considerable distance, is it not? So to make that signal go from your brain to your big toe as rapidly as possible, we want to be sure that those axons in the peripheral nervous system are myelinated. So that's typically why we talk about it within the context of the peripheral nervous system. Okay. All right, folks. So we'll wrap this topic up by defining the second type of cell within the peripheral nervous system. So we've defined Schwann cells. We also need to talk about the satellite cells. Satellite cells, take a look again at figure 12.7, and you can see on picture C as in Charlie, the satellite cell. And we won't spend too much time here. I'll just mention a couple quick things. First off, satellite cells surround the cell bodies of neurons that are part of ganglia, peripheral nervous system ganglia. These satellite cells provide a measure of structural support to these groups of cells. So there's a structure capacity to the satellite cells. And finally, satellite cells help regulate the exchange of materials between the cell bodies of the cells that are part of the ganglion and the surrounding interstitial fluid. So it acts as a go-between for material exchange between these cells and the extracellular fluid. Okay, so for our purposes in this course, Schwann cells are the big honcho, right? That's where I would want you to put most of your study efforts. Okay, folks, do you have any questions about the glial cells we've defined for the central nervous system or the peripheral nervous system? Okay, so let's continue. Now we are going to finally dip our toes into the water of electrical signal physiology. So I'm turning the page in the textbook. I'm looking at the section of the textbook where there's a section heading 12.3. It says electrical signals in neurons and overview. And a nearby figure for that would be figure 12.10, figure 12.10. All right, so I'm going to just give you a couple quick starting point background pieces of information, and then I'm going to show you some videos here. All right, so let's see. This probably, you can probably justify going down onto a new page here for notes. All right. So depending on how deeply you've read into chapter 12 or what experience you have from other biology courses, you know that in our nervous systems, there are basically two types of electrical signals. Many people have heard of nerve impulses and action potentials, but there is actually another type of electrical signal that maybe is not quite as famous. It's called a graded potential. So let's not get too far ahead, but there are two types of electrical signals in the nervous system. They are graded potentials. Okay. Now, when we say a graded potential, we're talking about an electrical signal that is used for very short distance communication. And by short distance, I'm talking about a couple millimeters. Okay, so hopefully you're a little bit familiar with the metric system. You know, if you happen to have a ruler nearby, typically on one side of the ruler, it shows you the inches, and on the other side, it shows you centimeters. And the centimeters are marked down in even tinier subdivisions called millimeters. Okay, so millimeters is what I'm talking about here in terms of distance, used for short distance communication. Okay. So when we talk about short distance, we're talking about probably, you know, maybe the width of a typical 10-cent coin we call a dime. I mean, that would be the kind of distance that a graded potential is used to transmit information. Okay. The second type of electrical signal, which is a little bit more famous, is the action potential. Action potentials are used for long distance communication, right? So if we think about a neuron that is starting at your hip and running down your entire leg to your foot, you know, that axon could easily be over 3 feet long, right? So we have some very, very tall people with long legs. So we could easily be talking about an axon that needs to spread an electrical signal over the distance of 1 meter or more. Okay. Upwards of 1 meter or longer. Okay, so let's see here. I'll give you 2 more ideas, then we'll get a video rolling. Remember, when an electrical signal is generated in the nervous system, we call it a nerve impulse. But when it's generated in a muscle fiber, it's called a muscle impulse. Or alternatively, when an electrical signal is generated in the nervous system, we call it a nervous system action potential, a neuron action potential. But when that electrical signal is generated in a muscle fiber, we call it a muscle action potential. So the name we give to an electrical signal is contextual based on the type of cell creating the signal. Okay. Neurons generate neuron action potentials. Myocytes, which is to say a muscle fiber, which is to say a muscle cell, generate muscle action potentials. Okay. So a muscle fiber generates a muscle action potential, a neuron generates a neuron action potential. Okay. Are there some subtle differences between the electrical properties of signals generated by neurons and muscle cells? Yes, there is. There are some subtle differences. But for right now, we're not going to focus on the differences. We're going to focus mainly on the main idea of what an electrical signal is, and we're going to focus on our conversation here in the nervous system about what these things are. Okay, so also remember that action potentials are also referred to as impulses. So we could have a nerve impulse, we could have a muscle impulse. Okay, just different ways. What I'm trying to say to you, these are different ways of basically referring to the same thing. Okay. All right. One final piece of information here. We need to talk about membrane potential. Membrane potential. Okay. Membrane potential is a catchphrase. It's a vocab word. It is a phrase which refers to a separation, or you might say a difference in electrical charge on 2 sides of a membrane. Remember, we talked about at the neuromuscular junction, we talked about the voltage-gated calcium channels. And we tried to say that the spread of the electrical signal down the axon bound to amino acids in the protein, which is the voltage-gated calcium channel, and changed the shape of that protein. In other words, the electrical energy on the inside and outside of the membrane was changed by the arrival of the electrical signal, which triggers the opening and closing of the voltage-gated calcium channel. Okay, so the plasma membrane of all cells, but in particular neurons and muscle fibers, always has some measure of difference in electrical charge. And that measure of difference in electrical charge is the membrane potential. And we measure the membrane potential typically in millivolts, okay? So membrane potential refers to the separation or difference of electrical charge on two sides of a membrane. Obviously, the membrane we're interested in is the plasma membrane of the cell. So we're talking about the separation of electrical charge on the inside versus the outside of the plasma membrane of either a neuron or a muscle fiber. For right now, we're in the nervous system chapter, so that's kind of how I'm focusing the conversation on the nervous system. Membrane potential refers to the separation of electrical charge on two sides of a membrane. Okay, that's going to be the plasma membrane. Membrane potential is typically going to be measured as in units of millivolts. Okay. One last talking point here. Okay. Let me ask you a silly question, but hopefully one that will help get you thinking about this in the chat window. Here we go. Will you typically set your parking brake in your car while on a shallow hill or a steep hill? Okay, a steep hill. Okay, please understand that is not a yes or no question. That is asking you to pick a choice, right? Pick one of the options. Okay. So obviously steep hill is the answer. And so here's what I'm trying to get you to see is that with membrane potential, the greater the difference, the more likely ions are going to move across the membrane. The greater the difference in charge, the more steep the hill, right? The more steep the hill, the greater the distance in charge, the more likely the car will roll down the hill, the more likely the ions will move across the membrane, okay? The greater the difference in membrane potential, the greater the greater the difference in electrical charge on both sides of the membrane, the more likely ions will flow across that membrane, okay? The greater the difference in charge across the membrane, the more likely ions will flow across that membrane from the area where you have a bunch of charge to the area where you have lesser charge, right? Okay, so with these ideas in mind, folks, I think we'll watch the first video here, okay? So let me get this queued up. And let's see. Yes, this is the first one we're going to do. Okay, folks, sit back and enjoy this. You don't have to take notes now because you know you can watch this again at home. Just watch it and try to take in what it's trying to say to you. Okay, here we go.
Membrane potentials. Graded and action potentials. In order to describe the generation of changes in membrane potential, we will use a motor neuron, which stimulates skeletal muscle to contract. The neuron can be divided into three zones. The input zone, consisting of the cell body and extensions known as dendrites, has chemically gated channels in its membrane to receive stimuli from other neurons as graded potentials. These graded potentials move toward the trigger zone at the axon hillock. The trigger zone is where voltage-gated channels are first encountered in the neuron. If the graded potentials reach threshold in the trigger zone, then an action potential is generated. The action potential is transmitted via voltage-gated channels down the axon to the output zone, where it causes skeletal muscle to contract. In the input zone, graded potentials are generated when stimuli such as neurotransmitters attach to receptors of chemically gated ion channels, causing them to open briefly. Then, shift in the distribution of ions on either side of the membrane results in a slight change in electrical potential. Graded potentials travel a short distance through an input zone toward the trigger zone before fading away. It is the net voltage produced by graded potentials at the trigger zone that determines if the threshold of voltage-gated channels is reached to trigger an action potential. Some graded potentials occur when chemically gated sodium channels open to allow sodium ions to diffuse into the input zone. This electrically moves the trigger zone closer to or above the threshold. They are considered to be depolarizing graded potentials. If chemically gated potassium channels in the input zone are stimulated to open, it allows potassium ions to diffuse out of the cell and causes the inside of the membrane to become more negative, moving the voltage in the trigger zone away from the threshold. These inhibiting graded potentials are known as hyperpolarizing graded potentials. Since depolarizing graded potentials cannot individually reach threshold, an insufficient quantity does not cause a response at the trigger zone. If, on the other hand, there are enough of them to reach threshold, an action potential is generated. If there are more hyperpolarizing graded potentials than depolarizing, the net membrane potential in the trigger zone becomes even more negative than the resting potential. This would temporarily inhibit the generation of an action potential or require additional positive stimulation to reach threshold. When the net graded potentials arriving at the axon hillock reach threshold, the action potential begins. Both sodium and potassium voltage-gated channels are stimulated to open. Sodium channels open quickly, allowing sodium ions to diffuse in and add positive charges to the inside of the cell membrane. If enough positively charged sodium ions diffuse in to bring the voltage on the inside of the membrane to 0 millivolts, it becomes uncharged, and we refer to the cell membrane as being depolarized. Sodium channels are only open long enough to allow sufficient amounts of sodium ions to pass through to cause depolarization. At this point, the inactivation gate on the sodium channel closes to block any further inflow of sodium ions. In actuality, before the sodium channels close, enough sodium ions diffuse in to cause the inner surface of the cell membrane to become positively charged. The purpose of the inactivation gate is to prevent the membrane from stimulating itself and causing repeated action potentials. At the same time the sodium channels close, the slower voltage-gated potassium channels open and allow potassium ions to rapidly diffuse out of the cell. The removing of positive charges from the inside of the membrane causes its internal surface to become negatively charged again or repolarized. Once enough potassium has diffused out of the cell to cause the inside of the membrane to be below threshold, the inactivation gate of the sodium channel opens as the gates on both types of ion channels close, but not before the inside of the membrane becomes less than the resting potential of minus 70 millivolts to cause the inside to become hyperpolarized. Sodium-potassium pumps transport sodium ions out of the cell and potassium ions into the cell to restore ion concentrations and to, along with leak channels, re-establish its resting membrane potential. The depolarization of one area of the cell membrane causes neighboring voltage-gated sodium and potassium channels to reach their threshold. The result is the action potential spreads like a wave across the entire cell membrane. Once the action potential begins, all of the output zone will depolarize and repolarize. This is known as the all or none principle. In other words, the action potential does not diminish as it travels across the membrane, unlike the graded potential. So either all of the membrane depolarizes and repolarizes, or none of it will. It is also important to note that the amplitude of the action potential is always the same. It is the frequency of the action potentials that signifies a stronger response.
Okay, folks, before I turn this off, I want you to appreciate something from our conversation this morning. In the video you see still on your screen in front of you, I'm going to reverse the video by about 30 seconds. Okay. And do you notice that there's these blobs of red ions flowing? Okay. 1, 2, 3, 4, 5, 6. Okay. Do you notice that these blobs are flowing everywhere along the axon, that you don't have one of these translucent ellipses? These translucent ellipses are Schwann cells. So this whole thing here is a myelinated axonal fiber. And what I'm trying to highlight to you is the action potentials along this axon are only happening in areas where you do not have myelin sheath, where you do not have a Schwann cell. So what I'm telling you is the video showed you, but did not tell you, that what you are literally witnessing right here in the video is saltatory conduction. This is saltatory conduction. This is passing the basketball down the court to the next teammate, and then the next teammate passes it to the next teammate, and then the next teammate passes it to the next teammate, and so on and so forth. So watch this again with an idea of saltatory conduction. Here we go.
All of the output zone will depolarize and repolarize. This is known as the all or none principle. In other words, the action potential does not diminish as it travels across the membrane, unlike the graded potential. So either all of the membrane depolarizes and repolarizes, or none of it will. It is also important to note that the amplitude of the action potential is always the same. It is the frequency of the action potentials that signifies a stronger response.
Okay, so there you have it, folks. In 6 minutes, you now understand the physiology of a nerve impulse, right? It's about as clear as mud at this point, I suspect, right? I mean, this is a heavy topic, so we're going to have to pound on this the rest of today and next class period to try to get this across. Are there any questions you'd like to ask at the moment, having seen the video one time and while I get the notes ready to take again? Okay, Amy, I understand that. So Amy says, even after the video, it still seems confusing. I get that. This is a heavy topic, and it's going to take quite a few hours of dedicated effort and study to peel back the petals of ignorance and confusion. And I'm going to help you the best I can here with our time. All right. So let me give you more background. Let's talk about it some more. And then I'll show you another video. We'll talk about it some more. We'll keep doing this over and over again with the time we have available. And then I'd like you to go back and watch more videos and study on your own during your private study time. And it will slowly come into focus, okay? All right, so we've talked about electrical signals, graded and action. We've defined kind of what they are. The video tried to show you the physiology of these things, and that's what we need to understand in greater detail at this time. Okay. And we also talked about this membrane potential idea. In other words, there's a typical difference in electrical charge on both the inner and outer surfaces of a membrane. That is the membrane potential. That difference is the membrane potential. And that membrane potential is measured in millivolts. And you noticed in the video, they had a graph where they were trying to show you how the millivolts were changing as ions moved back and forth across the membrane. So let's continue here. Please ask questions along the way. All right, number 4. So critical in the video was these ion channels, right? Particularly it harped on these sodium ion channels that opened up, allowing sodium to enter the cell. That was depolarization. When you depolarize a neuron, you make the inside momentarily more positive than it had been a second ago. That is the trigger for an action potential. When you allow potassium ions, which are stored inside the cell, back out, you are removing positive charge, which means you are returning the neuron to a more resting state. That's called repolarization. Don't worry, we're going to get these things into your notes. So let's talk about these ion channels a little bit. Okay, ion channels, as you know from our conversation with the neuromuscular junction, are proteins. They're proteins that have a tunnel inside the dimensions of the protein that allow ions to move back and forth across the membrane down what we call the electrochemical gradient. Ion channels are proteins that have tunnels inside them for ions to move through. Okay, when the protein is embedded in a membrane, like the membrane of the neuron or the membrane of the muscle cell, when that protein's embedded in the membrane, that channel provides a way for an ion to either enter a cell or exit a cell. Okay, so let me just read my own notes. Sometimes I type too fast and I bugger it up. Ion channels are proteins that have tunnels inside them for ions to move through. When these proteins are embedded in the membrane of a neuron or muscle cell, the tunnel inside the protein provides a way for ions to enter or exit a cell. Okay, ions will move down. Ions tend to move down their electrochemical gradient. So let's say it this way: ions move down their electrochemical gradient. Okay, and so notice that that phrase electrochemical gradient basically has two ideas in: electrical, and chemical, meaning the actual atom, or in some cases a molecule. Let's talk a bit more deeply about this. The electrochemical gradient refers to a gradient. And so you say, well, okay, great. What is a gradient? A gradient is an area of difference, an area of difference. Think about you could be walking someplace and you start walking up a hill. You start walking up a hill. And then once you reach the top of the hill, you start walking down the hill. So if you think about your position at the bottom of the hill and the top of the hill, the bottom of the hill and the top of the hill, that difference in elevation is a gradient. That difference in elevation is a gradient. The bottom of the hill, top of the hill, the difference in elevation is a gradient. Now, we're not talking about an elevation gradient here with our physiology. We're talking about an electrochemical gradient, okay? So an electrochemical gradient is an area of difference that has two aspects, one electrical and the other chemical. Okay, so let me give you an example of how this works. Okay, if on one side of a plasma membrane we have 100 sodium ions, which have positive charge, right? If we have 100 sodium ions on one side of a membrane, and on the other side of the membrane, we have 25 sodium ions, okay, so let's say on the sodium ions with 100, the side of the membrane with 100 sodium ions, we'll call this side A, just so I can refer to it. And the other side with 25 sodium ions, we'll call this side B, okay? So the area where you have 100 sodium ions is side A, the area with 25 sodium ions is side B, okay? In this kind of scenario, hopefully it is clear to you that the sodium ions on side A are going to move towards side B, not the other way, not the other way. The area with greater positive charge and greater number of ions, which is to say atoms, is going to have those atoms, those ions, move to the area where there are fewer. So we're going to have some of the 100 sodium ions in side A start to migrate across the membrane to side B, okay? Okay. And the reason that the motion of the ions is moving from A to B and not B to A is twofold. First off, okay, so let's see here. The reason ions move from side A to side B is twofold, okay? First off, side A has more positive energy, more positive charge. That's the electro portion of the electrochemical gradient. Side A has more positive charge. Additionally, not only does side A have more positive charge, it also has physically more atoms. There are sodium atoms that happen to have a charge, so they're ions. So side A has more ions, it has more atoms. That is the chemical portion of the electrochemical gradient. Okay, so let's review this example here one more time. Let me read it. If we have 100 sodium ions on one side of a membrane that we're calling side A, and on the other side of the membrane, we have 25 sodium ions we're calling side B. In this scenario, some of the 100 sodium ions in side A will start to move across the membrane to side B. Why? Because side A has more positive charge. Side A also has more ions. than side B. So the more positive charge is the electrical portion of the electrical chemical gradient, and the larger number of ions / atoms is the chemical portion of the electrochemical gradient. Okay, so question in the chat window. Are you with me so far? Are you with me so far? Any questions? Now let's change this up a little bit. Okay, let's change this up a little bit. So this was example 1. Now let's come down here and change this up just a little bit. Okay. So for example 2, if we have the same situation that we have defined in example 1, but we add 25 potassium ions to side B, that's going to mix things up a little bit. Okay, so for example 2, assume we start with the same situation as example 1, and then we magically, okay, so we magically add 25 potassium ions to side B. Okay, so in this scenario, what's going to happen? Okay, what will happen and why? Okay, all right. So I'm going to punch down on the next page here, folks. Okay, what will happen and why? Well, here's what's going to happen. Some of the sodium ions from side A are still going to cross into side B, but they're going to be fewer sodium ions that move, and they're going to move with less energy because the gradient is more shallow at this point. It's like walking up a shorter hill rather than a larger hill. Okay. Some sodium ions on side A will still move across to side B, but fewer ions will move, and those ions will move more slowly because the gradient, the electrogradient, is no longer as steep as it was in example 1. Because the electrochemical gradient is not as steep as it was for example 1. So you ask yourself, well, why not? Okay, well, think about this. We have just in example 2 here, we have added 25 potassium ions that have positive charge, right? That has positive charge. So that means, okay, so that means, okay, I'm thinking about what to say versus what to type here. Basically, with example 1, here's what we're dealing with, and let me draw here, okay? With example 1, I have the membrane. Here's side A. Here's side B. I have 100 sodiums, 25 sodiums. Okay, so from an electrical perspective, I have 100 minus 25. The membrane potential, the difference is a difference of 75. So this is the steepness of my hill. Okay, that's the steepness of my hill. But in example 2, excuse me as I go down here, in example 2, here's what I got. Got the membrane again. I got side A here. I got side B here, right? I got 100 sodium ions here. I got 25 sodium ions here. And I've also got 25 potassium ions here. So from an electrical perspective, I now have this. And I'll do it in a neutral color, not to imply anything. I have 100 minus 50, right? So 25 and 25 is 50. I have 50 ions that collectively represent 50 something with a positive charge. So now my difference is 50. So now this is the steepness of my hill with example 2. So let me ask you in the chat window, which hill which hill, example 1 or example 2, has the greater membrane potential? Okay, thank you, Christian. How about the rest of you? Okay, Lauren, you say hill 1 because of how it's more steep. That's correct. Okay, so I get the idea that there's still a little bit of fuzziness here, right? Because you're slow to respond. So that's exact, so folks, in the chat window, my question was, which hill, example 1 or example 2, has the greatest membrane potential? Folks, I'm going to put another question into the chat window. And I'm going to tell you that the question I'm going to put in the chat window means the exact same thing as my first question, but I'm going to word it differently. So here it comes. And remember, this question, which is worded differently than the first question, means exactly the same thing. So what's my answer to the second question? Okay, it's hill 1. So folks, appreciating that the last 2 questions I put into the chat window mean exactly the same thing using different words. Is it getting clearer now? Because I hope you can see that the difference, right? Here's the difference, right? The difference, right? This is a minus sign, right? This is a minus sign. So the minus sign represents a difference. What is the difference in our conversation? This is the membrane potential. The greater the difference in membrane potential, the more likely ions are to move across the membrane, right? Didn't we put that up in your notes somewhere up here? Here it is. Crap. Right here is what I'm talking about, right? The greater the difference in charge across the membrane, the more likely ions will flow across that membrane. That's what I'm trying to get you to see. The greater the difference in charge, and that charge is a community charge. It could be 25 sodium atoms with a positive charge plus another 25 sodium atoms with a positive charge, or it could be 25 sodium atoms with a positive charge and 25 potassium ions with a positive charge. So you add up all the ions on one side of the membrane and determine how much positive or negative energy you have there. And then you subtract that from the same thing on the other side of the membrane. Whatever that magnitude is of that subtraction process, that's your difference in charge. That's your membrane potential. Okay. All right. Question in the chat window. Better or worse right now? Is it getting through? I know it's hard. Please ask questions. What can I do to try to help you understand this better? Okay. All right, folks, we are out of time. So unfortunately, I'm going to have to stop. But let me say that we're going to spend all next lecture on this topic. I mean, I am throwing you to the wolves, right? I've thrown you to the wolves all semester long because this is a hard course. But we have another full lecture where the whole lecture is going to be about understanding the electrical signal business, okay? So don't sweat too much. You're going to get some more help here. All right, so I'm going to stop at this time.